Hey guys, welcome. In our lesson today, we are going to discuss paper 1 of the TAP trial for exam. By the way, in this lesson of ours, we are going to tackle an interesting question on genetic engineering. Did you know that bacteria are being used for the mass production of insulin, which is then administered to people who are diabetic? Bacteria, yes. Sounds like science fiction, right? But it's actually true. So this process involves the use of plasmids, which are small DNA molecules present in bacteria. So if you wish to learn more, and you should, because the whole process is utterly fascinating, stay tuned and I bet it's going to make a fascinating lesson. Question number 1. State 2 distinguishing features of kingdom Monera, okay? So when we talk about distinguishing features, we mean features that are unique only to the members of kingdom Monera. And who are these? The bacteria, of course. So number 1, they are prokaryotic. In fact, this is the most fundamental characteristics that sets bacteria apart from any other organism belonging to the other 4 kingdoms. So bacteria are prokaryotic in that their nuclear material is not surrounded by a membrane. All the other 4 kingdoms have organisms whose nuclear material is surrounded by a membrane and is therefore referred to as eukaryotic. So number 1, prokaryotic. Another unique characteristic of kingdom Monera is that they lack membrane-bound organelles, okay? Now, when we talk about organelles, these are the small structures that are present within the cell. Most tend to be surrounded by a double membrane. For example, the mitochondria, the Golgi bodies, the lysosomes, and so on. Now, when it comes to bacteria have few organelles, and those that are present tend, not to have a double membrane. So they don't have these membranes. Number 3 is that they also have a cell wall that is made up of murine. Now, when it comes to cell walls are present in a lot of organisms. They are present in members of kingdom Protoctista. Some of them do have cell walls. They're present in members of kingdom fungi and, of course, in plants. Now, when it comes to bacteria, their distinguishing factor is not that they have a cell wall, it's that their cell wall is made up of a complex carbohydrate or a complex compound called murine. Murine is actually a compound that consists of complex sugars and protein. On to question number 2a. What is biotechnology? So biotechnology is a branch of biology that deals with the development of techniques for the application of biological processes. Moving on to part B. State 1 characteristic of living things found in animals but not in plants. Okay. Now, earlier on in Form 1, you were introduced to the 8 characteristics of living things. For a substance to be classified as a living organism, it needs to have all of these 8 characteristics. Now, what the question is asking us is that out of these 8, there is one that is absent, in plants, but is present in animals. And the only one is locomotion. Now, locomotion actually falls under movement. So movement can be either movement of the whole body, or you can have movement in terms of changing the position of one or more parts of the body. Plants are capable of movement when they fold their leaves, when they open and close their flowers, when their roots stretch deeper into the ground. All of these are different types of movement that are witnessed in plants. What plants lack, however, is locomotion. Locomotion is a type of movement whereby we are talking about moving the whole body from one part to another. This, of course, is absent in plants. Now, I want to say this. When you get such a question and you are asked for a characteristic, for you to state the characteristic, please just state it, okay? Don't go into details and explain Animals are capable of locomotion while plants, cannot do so. Just stick to the script, stick to what the instructions ask of you. Moving on to question number 3. Explain reasons for each of the following. Roman number 1. Tracheoles are the respiratory sites in insects, yet they are not vascularized. Okay. Now, what are tracheoles? Tracheoles are tubes that branch extensively throughout an insect's body. So these tubes start out from the spiracles. Spiracles are external openings that are found on the thorax and the abdomen of an insect. So the spiracles open up into the trachea. The trachea then divides repeatedly into smaller and smaller tubes, and these are what we call the tracheoles. So the tracheoles lie in very close contact with all the cells in an insect's body, okay? And that is the term we actually use for this is ramify. You might have seen it in Kelby. So ramify simply means that tracheoles, are found in every part of an insect's body, and therefore every cell is in close contact with a tracheole. Roman number 2. Alveoli do not collapse even when their volume decreases during exhalation. Okay. So according to this question, when we exhale, when we breathe out, okay, the alveoli tend to lose a lot of air because it passes out of our body through the nostrils. But even when that happens, they don't collapse completely. So what's stopping them from doing so? The reason is that there will always be residual volume of air left behind. Okay. So this essentially means that even with your most forceful exhalation, there will still be a small amount of air that will remain in the alveoli. And this is what is termed as the residual volume. So this ensures that the alveoli are never completely empty of air. So having this residual air inside the alveoli prevents them from completely collapsing. Moving on to question number 4. The diagram below shows a living organism. Okay. Part A. State the class to which the organism belongs to. Now this is a millipede. Millipedes belong to class Diplopoda. A reminder, when you are writing the name of any taxa, you should always start with a capital letter. Part B. Give 2 reasons for your answer. Okay. Number 1, we have a cylindrical body, right? Okay. 2 pairs of legs per segment. You can also go ahead and state that its body is divided into 3 parts. Now, I want to say this. If you decide to choose this point, then you cannot leave it as such. You cannot write it as 3 body parts. No, you have to state what the body parts are. So you have to state that the body is divided into 3 body parts. Yes, the head, a short thorax, and of course the trunk. And yes, specify that the thorax is short. Moving on to question number 5. Below is a diagram of a plant cell. Okay, let's take a few seconds to look at it. It's a typical plant cell. Part A. Label structure A and structure B on the diagram. Okay, let's look at A. A is clearly the nucleus, okay, and B, that is our sap vacuole. Please, when you are, identifying the vacuole that is present in a plant cell, ensure to include the term sap vacuole. Because when it comes to vacuoles are actually of 3 types. We have the sap vacuole that is specifically found in plant cells, and then we have the contractile and food vacuoles. These are present in unicellular organisms. Now, I want to say this. Part A was very specific on where the answers were supposed to be. So as you can see, after the labels A and B, you are provided with a space where you should write down your answer. Some students might ignore this and write down the answer at this point. Please don't do so. If you do so, you're not following the letter of instructions and will be penalized. Part B. Give one difference between an animal cell and the plant cell shown in the figure. Now, this is a very easy question. As you are aware, there are several differences that are present between a plant cell and an animal cell. So you can choose the easiest one that you remember and just write it down. So any of those is correct. Okay. Moving on to question 6. Explain why the body temperature on a healthy human being may rise up to 39 degrees Celsius on a hot, humid day. Okay. So when it comes to humans are endotherms, and we usually maintain a core body temperature of around 37 degrees Celsius through a process called thermoregulation. Okay. Now, this balance is achieved by equating production of heat with heat loss. So we lose some heat, we gain some, through metabolic processes. And at the end of the day, through this, we maintain a body temperature of roughly 37 degrees Celsius. This, of course, is the optimum temperature for, metabolic activities in our body. Now, on a hot day, there is going to be a significant amount of heat in the surroundings. Now, one of the ways our body loses heat is by evaporation of sweat. When it evaporates, it leaves a cooling effect on the body, okay, because it uses heat from our body. However, on a humid day, this process is going to be affected because the air will already be saturated with water vapor. This means that the concentration gradient between the skin, which will already be covered with sweat, and the surrounding air is going to be low. So you're going to have sweat on the surface of our skin. You're going to have air that has a high humidity. It has a lot of water vapor in there, okay? Remember, we were told that the day is hot and humid. So you're going to have less evaporation taking place. So cooling of the body will slow down. So if the body, if cooling of the body slows down, and already we are talking about this being a hot day, what will happen is that you are going to have the body temperature rising because the heat has nowhere to go. So the temperature of the body will increase up to 39 degrees Celsius. Part B. In an experiment, a piece of brain was removed from a rat. It was later found that the rat had large fluctuations of body temperature, suggests the part of the brain that had been removed. Okay, so a part of the brain of the rat was removed. And apparently, this will be the part of the brain that is involved in thermoregulation, in controlling the temperature of the body. Why am I saying so? Because with its removal, the rat experienced fluctuations, changes, erratic changes in the body temperature. So, the part of the brain that was removed will definitely be the hypothalamus, because this is the part of the brain that is responsible for thermoregulation. Moving on to our next question. The diagram below refers to the plasma membrane of an animal cell. Okay, so plasma membrane is simply the cell membrane. So if we look at this diagram of ours, we can clearly see that there are two processes taking place. We have X and we have Y. Okay, so the movement is happening in opposite directions, and it's actually of two materials. We have glucose being undertaken in process X, oxygen in process Y. Part A. Identify the two processes X and Y and explain your answer. Okay, so let's start with X. If we look at X, we are having glucose moving from a region of low concentration to a region of high concentration. What is that? That is active transport. Why? Because movement is against the concentration gradient. We are talking about particles, specifically glucose, moving from a region of low concentration to a region of high concentration. What about Y? So we are having oxygen moving from a region of high concentration to a region of low concentration. High to low. That is diffusion. Reason being, movement is along the concentration gradient. This simply means that it's from a region of high concentration to a region of low concentration. Which of the processes X and Y will be affected by the presence of metabolic poison in the cell? Explain. Okay, first let's start with this. What is a metabolic poison? So these are substances that interfere with the normal activities that happen within the cell. So usually they tend to target enzymes that are involved in respiration, okay, or other components involved in respiration. So these are chemical substances that simply interfere with the process of respiration. And if they interfere with the process of respiration, that means they will also be affecting active transport. This is because active transport relies on the production of energy. So if you have a metabolic poison, that means respiration will be affected. If respiration is affected, then that means active transport will also be affected. So which process will be affected? X, because this requires energy. Remember, diffusion is a passive process. That means it doesn't require energy. Any process that involves movement of particles along the concentration gradient, from a region of high concentration to a region of low concentration, then definitely does not require energy because this movement occurs naturally by itself. Moving on to question number 8. Give 2 reasons why Latin is the most preferred language in binomial nomenclature. Okay. Now you might have noticed that when it comes to naming, the scientific naming of organisms, we use Latin language. Now this is for several reasons. Number 1 is that this language is static. It does not change over time. Reason being that Latin is a dead language. A dead language is simply one that is, not a primary language used by people for communication. Therefore, if it's not used by people for communication, it will not change over time. Okay? So no new words are added, no words are removed. It's just going to remain the way it is because it's not really being used as a means of communication. So it's static. Sometimes students might write that it's not dynamic. It's simply the same thing. Okay. Now, another reason is that this was also the language that was used by scientists at that time. So a lot of the scientists were based in Europe at that time, and most of them used Latin as a method of communication. So it was natural for them to name organisms based on that language. Question number 9. When placed into a beaker of water, a red blood cell bursts, while a plant cell does not burst. Explain why the red blood cell bursts, but the plant cell does not burst. Okay. Now, this is a straightforward question. If you were to take a plant cell and an animal cell, which in this case is the red blood cell, place them in distilled water, okay? What will happen is that the plant cell, actually both of them, will take in water. The difference is that even with gaining of water, the plant cell never bursts. Reason being that it has a very rigid cellulose cell wall. So on the other hand, an animal cell will take in water and eventually burst. Reason being that it lacks a cell wall. It just has a cell membrane which is flexible. So if it takes in too much water, it will later on burst. Question number 10. In an investigation, a student collected two plants, A and B. Plant A had hairy leaves and few stomata, okay, which were sunk into the epidermis. Leaves of plant B were broad and had many stomata on the upper surface only. In which habitat would you find plant A and plant B? Okay. Now, if we look at plant A, it's clear that it has adaptations that are there to minimize water loss. Okay. Talking about hairy leaves, which will trap moisture, few stomata, and these stomata are sunken. These are adaptations that are found in plants that are present in deserts, arids, or semi-arids area. Any of this is correct. Plant B, on the other hand, has broad leaves, many stomata, and these stomata are located on the upper surface. Actually, only on the upper surface. So this is an adaptation of an aquatic plant. Question number 11. The diagram below represents a section through a chloroplast as seen under the electron microscope. Okay, let's briefly look at the structure. By the way, if at any point you need more time to look at the diagram or to go through the answers by yourself, kindly pause the video and do so. Part A. Name the structure labeled D. Okay, let's look at where D is. Okay, that's clearly the outer membrane. A chloroplast, just like with a lot of the organelles, tend to have a double membrane. Okay, so this is the outer membrane. Part B. In which of the labeled structures would you expect to find chlorophyll molecules? Chlorophyll molecules are present in grana. Okay, but A is pointing at one, so that is the grana, right? Now, if you have such a question, it doesn't want you to provide the name. Why? Because you have been specifically told in which of the labeled structures. So it wants you to give any of the labels assigned. So that is going to be A. Part C. In which of the labeled structures does carbon 4 oxide fixation occur? So carbon 4 oxide fixation occurs in B. Specifically, it occurs in the stroma. Now, if you were to write the name itself, you would be penalized, okay? So when you go through such questions, please always pay, very close attention to what the questions requires of you. Moving on to question 12. An investigation was set up to demonstrate the response of flatworms to the presence of food. A piece of liver and a glass bead were placed in a dish. 15 flatworms were then scattered randomly into the dish as shown in the diagram below. Okay, so we have the liver on one end, the bead of glass on the other end, and we have the flatworms scattered into the petri dish. After 30 minutes, it was found that there were more flatworms near the liver than the glass beads. Okay, so it's clearly that the flatworms were moving towards where the liver is. Part A. Account for the observations. Now, the movement of the flatworms is based in chemotaxis. Okay, specifically positive chemotaxis. This is the movement towards chemical substances. The liver contains proteins, carbohydrates, lipids, all of which are chemical in nature. Hence, the liver provides a chemical stimulus. So the flatworms moving towards where the liver is positive chemotaxis. Now you might be thinking, okay, what about the glass bead? The glass bead is inert. It does not emit, it does not produce any chemical signals equal to. So you might be thinking, Sawa, then what is the purpose of including the glass bead in this experiment? Simple. It acts as a control. Since it is an inert object, it does not release any chemicals. Therefore, the movement away from the beads towards the lever shows that it's specifically the chemicals in the lever that are influencing this movement. Part B. State the purpose of the glass beads. As stated, this is, it's a control. Yeah. Aya, tunaendelea. Question 13. The diagram below shows a germinating leguminous seedling. Okay, let's take a moment to look at the diagram. So we have the seedling as it germinates in 3 stages. Okay, part A. Other than the emergence of cotyledons above the ground, give a reason why this is apical germination. Okay, reason is that the hypocotyl elongates faster than the epicotyl. Part B. State the stimulus responsible for straightening of the part labeled H. That is the shoot. The stimulus responsible is light. Question number 14. Part A. What are vestigial structures? These are structures that have ceased to be functional and in the course of time have become reduced in size. Now, I want to say this. Sometimes students confuse the two. They could state that these are structures that have become reduced in size and have stopped functioning. No. These structures stopped functioning, and because they no longer played a vital role in the organism's life, they have then become reduced in size. So it's one after the other. Because they are no longer useful, they then became reduced in size. Part B. With reference to a shark and a whale, describe convergent evolution. Now, sharks are fish, right? They belong to class Pisces. Whereas whales are mammals and therefore they belong to class Mammalia. So you can clearly see we have 2 organisms that belong to different classes. Now, despite both of these having different ancestry, belonging to different classes, they have evolved features that have adapted them for efficient movement and survival in aquatic habitats. Now, a good example of such a feature is the presence of fins. Okay, both have fins and they use them to propel themselves through water. Now, while both of them might have fins, which may look similar, and perform similar roles, the underlying structures that are present in these fins are entirely different. Okay? And because of this, we have such structures which we refer to as analogous structures. Now, analogous structures are present as a result of convergent evolution. So this is a type of evolution where you're going to have analogous structures modified to perform similar functions for the purpose of exploiting the same ecological niche. So in this case, they both have fins, okay? But because they have different ancestry, the structures of the fins themselves are completely different. So they have a shallow resemblance, but are different. And these are referred to as analogous structures. Moving on to our next question. The diagram below shows an experimental setup using soaked bean seeds. Okay, let's take a moment to look at our diagram. So we have a retort flask containing soaked bean seeds. On the neck of the flask, we have cotton wool, above which is a stick of potassium hydroxide. The flask has then been immersed in a beaker of colored water. So we are supposed to state the aim of the experiment. The aim is to show the respiration of germinating seeds. Part B, account for the results at the end of the experiment. Okay, so with this question, we are supposed to state the results at the end of the experiment and then explain how they came about it. Now, what are the results? The colored water will rise up the flask. Okay, how did this happen? Number one, soaked beans will start to germinate. Remember, if you have conditions such as enough oxygen, water, you are going to end up having germination. These are seeds that have been soaked. So germination will take place. Now, germinating seeds are metabolically active. So they are going to carry out cellular activities and therefore they need energy. This energy will be produced through aerobic respiration, where the seeds will take in oxygen from the surrounding and then, release carbon dioxide. So they will take in oxygen, use this oxygen in the process of aerobic respiration to break down their food substances to provide them with energy. Now, respiration uses up oxygen and releases carbon dioxide. Sawa? So the oxygen within the flask will be used up during aerobic respiration and carbon dioxide will be produced. However, we have a potassium hydroxide stick in the flask. So what will happen is that this will absorb any carbon dioxide produced by the seeds. So you're going to have a case whereby oxygen has been used up during respiration. Carbon dioxide produced has been rapidly absorbed by the potassium hydroxide. So this will cause a decrease in the volume of gases within the flask. And eventually it will cause the colored water to rise up the tube. Okay. Moving on to question number 16. State the differences between the composition of maternal blood entering the placenta and maternal blood leaving the placenta. Okay, so entering the placenta, the maternal blood is going to have a high concentration of oxygen. Okay, it's also going to have a low concentration of carbon dioxide and any nitrogenous waste, and it's also going to have a high amount of nutrients. Okay, these of course are going to be supplied to the fetus. Now, leaving the placenta from the fetus to the maternal blood is going to have a low concentration of oxygen. Most of the oxygen will diffuse across the placenta and into the fetal blood. So the blood leaving the placenta is going to have a low concentration of oxygen, a high concentration of carbon dioxide and nitrogenous waste, and low concentration of nutrients. Question number 17, part A. Name the causal organisms for each of the following diseases. Okay, for malaria, that is Plasmodium. Now, Plasmodium has 4 species, so you can write any of the 4. For example, Plasmodium vivax, malariae, and so on. Just a reminder, when doing so, don't forget the rules of binomial nomenclature. The generic word is supposed to start with a capital letter. And because it's handwritten, you are supposed to separate these two with a line. Okay, you're supposed to underline separately. Now, Roman number 2, typhoid, that is Salmonella typhi. Part B. Explain why patients suffering from amoebic dysentery tend to have bloody stool. Okay, bloody stool simply refers to bloody feces. Now, when it comes to amoebic dysentery, the infection usually begins when a person ingests cysts of Entamoeba histolytica. Okay, this can be done through contaminated food or water. So these cysts are then going to move to the small intestines where the covering are going to break open, releasing the protozoa, which is now going to be active. These will then move onto the large intestine. Now, when they reach the large intestine, they are going to stick to the walls of the intestine and start secreting enzymes that break down the wall of the intestine. This, of course, will lead to the formation of ulcers. Now, sometimes the ulcers can penetrate deep into the intestinal lining, causing damage to blood vessels, okay, such as capillaries. Now, when these blood vessels burst, they release blood, which is then passed out in stool. Question number 18. State two functions of the rib cage and the stomach. Okay, so to protect vital organs such as the heart and lungs. Now, I want to say this. When you state this point, please do not leave it as such, protect vital organs. We have quite a few vital organs in our body, and not all of them are protected by the rib cage. For example, the brain. You cannot mention it in general, like protect vital organs, and include them all under this point. I hope I'm making sense to you. Anyway, when you're writing this point, kindly specify even one organ, so such as the heart or the lung. Number 2, they hold the internal and external intercostal muscles, which facilitate breathing. So yeah. Part B. Name the type of muscles found in each of the following parts of the human body. So we have the uterine wall and the fallopian tubes. Now, when talking about muscles, we only have 3 types of muscles. Number 1, we have the cardiac muscles. These, of course, are only found in the heart, so that takes them out. We also have the skeletal muscles. Skeletal muscles are those that are attached to our skeleton. If you look at the uterine wall, Yamanisha, it's in the uterus, okay? The fallopian tubes, those are not attached to the skeleton, so that takes out the skeletal muscles too. And which are we remaining with? These are the smooth muscles. Smooth muscles are also known as visceral muscles. So these are present in both. Moving on to our next question, the graph below shows the curvature of the human lens when viewing objects at different distances. Okay, let's take a moment, look at the graph, pause the video if you need more time. So over here, we have a bar graph that is related to the curvature of the eye lens. Now, this information is related to a process called accommodation. What is accommodation? This is a process whereby changes occur in the structures of the eye to ensure that the light rays from the object at different distances are sharply focused onto the retina. Okay, simply put, this is a process that enables us to focus on objects that are at different, distances from us. So through the process of accommodation, we can focus on objects that are far, those that are near, and so on, ensuring that we have a sharp image of them. Accommodation. Now, in order for us to focus on objects that are far, the lens becomes thinner and less curved as such. This is because light rays from distant objects have traveled over a long distance before reaching the eye. So in order to focus these rays onto the retina, the eye lens needs to be its thinnest and least curved, okay? To ensure that the light rays can then be refracted onto the retina and not before it. Now, when we focus on a near object, the light rays from the object have traveled a short distance, okay, because the object is near. So in order to focus these rays onto the retina, the lens becomes thicker and, more curved, okay? Such a lens has a greater refractive power, meaning that it bends the light rays more. Why is it bending the light rays more? To ensure that the rays are, refracted precisely onto the retina and nowhere else. Okay, so allowing you to clearly see the object. By the way, you can just put this into practical. Okay, into practice. How do you even put that in English? Anyways, if you were to look at an object that is far, there's going to be just a few seconds where your eye focuses on the object. The image seems blurry before it turns sharp. Same goes for a near object. So that short period of time before you get a sharp image is where accommodation comes in. Now, if you look at our question, the lens that has the highest curvature will be the one from the nearest object. Remember, when we look at objects that are near, the lens becomes thicker and more curved, okay, to bend the light rays onto the retina. The nearer the object, the more curved the lens will be. So, therefore, which one of the above objects is closest to the human eye? We are going to look at the object that has the highest curvature, okay? And that is number 2. By the way, number 2 and number 4 are quite close, but number 2 takes the cake. Part B, explain your answer. So the lens has the highest curvature in order to increase the refractive power so as to focus the light rays onto the retina. Moving on to question number 28. Give 2 examples of vestigial structures. Okay, now we have several vestigial structures in different organisms. So for example, in the case of humans, we have an appendix. We also have the coccyx in humans. We have the nictitating membrane in mammals, although nictitating membrane is present in some mammals. Yeah. Anyway, I want to say this. When it comes to vestigial structures, you need to specify where they are. For example, when I talk about the appendix as a vestigial structure, it's in humans, not in all organisms. For example, when it comes to ruminants such as cows, they make very good use of the appendix, but not in humans. So specify the organism where the vestigial structure is found in. Part B. Explain the evolutionary relationship between the fin of a fish and the flipper of a whale. Okay, this question has actually been repeated. It's similar to what we discussed before. So I'm just going to go straight into the answer. Now, these 2 organisms have different embryonic origins, but have structures, okay, which are the fins, that have been modified to perform the same function, which is swimming or locomotion. Question number 21. State 2 differences between epigeal and hypogeal germination. Now, in epigeal germination, the cotyledon emerges above the soil. In hypogeal germination, the cotyledon remains below the ground. In epigeal germination, the hypocotyl elongates faster than the epicotyl, while in the hypogeal, it's the epicotyl that elongates faster than the hypocotyl. Okay, question number 22. State 2 adaptations of Schistosoma mansoni to the parasitic way of life. Now, this is the organism that is responsible for bilharzia, and it has quite a few adaptations, okay? So I'm just going to mention a few of them, and here they are. Moving on to question number 23. The diagram represents a simple reflex arc. Part A. Account for the appearance of the part labeled S. Now, when it comes to S, X is a section of the spinal cord, but specifically that is the white part of it, okay? Now, the reason why it's white in color, if you look closer at it, you're going to note that it has quite a few axons from the neurons that are present, okay? Now, axons are surrounded by a myelin sheath. The reason why they are covered by a myelin sheath is for, faster transmission of impulses. Now, myelin sheath is made up of a fatty substance. Fats tend to be lighter in color and also shiny, and that is the reason why the part labeled F tends to be white in color, because it consists of axons of the sensory and motor neuron, which are myelinated, okay, or which have the myelin sheath. Part B, name the enzyme involved in the transmission of impulses between P and R. The enzyme is cholinesterase. Part C, state one structural difference between P and R. Okay, let's look at where P is. That is our relay neuron. And R, that is going to be our motor neuron. So when it comes to P, it lacks the myelin sheath. Okay, so relay neurons are not covered by the myelin sheath. So this is non-myelinated. While R, of course, is covered by a myelin sheath. Now, P is bipolar, R is multipolar. So any of these two is correct. Question number 24, part A. The white eye color in Drosophila, these are the fruit flies, is caused by a recessive sex-linked allele. Let's pause there. We have been provided with two bits of important information. Number one is that the allele that causes, that is responsible for white eye color is sex-linked, meaning that it's present in the sex chromosomes. Which are the sex chromosomes? The X chromosome and the Y chromosome. Now, when it comes to this particular allele, it's only present on the X chromosome. Now, we know that when you have a crossing and it involves alleles that are present on the sex chromosomes, then when you do, your crossing with the genotypes, it has to include the sex chromosomes themselves, X and Y chromosomes. Now, another important information that we've been provided with is that this allele is recessive, okay? So we're being told that dominant allele, capital R, is for red eyes. What result will be obtained from a cross between a white-eyed female and a red-eyed male? Okay, so let's start. Our phenotypes are as such. The female is white-eyed and the male is red-eyed. Now, when it comes to the genotype, a female has two X chromosomes. This allele is present on both X chromosomes, okay? So we're going to represent it in both. Now, remember, this allele is recessive. So it can only be portrayed in the phenotype if, in this case of the female, it's present on both X chromosomes. Therefore, since it's present in both X chromosomes, na tumejua iko present because the female is white-eyed, okay? Aya. What will happen is that we are going to have it on both. And we are going to use a small r, okay? Because the allele is recessive. And we always use small letters for those. Okay. Now, in the case of the male, it has two sex chromosomes, an X and a Y. There's not going to be anything on the Y chromosome. On the X, we are going to have a capital R. Why? Because the phenotype of the male is red-eyed. So that means it has the dominant allele. So once we have our crossing, and there we go. So that is going to be our result of the F1 generation. Now, just for fun's sake, let's look at the genotypes. So with the first female, that is going to be red-eyed, okay? So it has one allele for red eye, one for white eye. But because the allele for red eye is dominant, it's the one that will be expressed. So it's going to be red-eyed. The other is, actually, they're both the same, okay? They are both heterozygous, and they will both be red-eyed. When it comes to the males, both males are going to be white-eyed. Okay. B part 1. State 2 characteristics of polyploid plants. They tend to have high yields, resistance to drought, resistance to pests and diseases, early maturity. Yeah, good thing. Haya, tunaendelea to our last question, and that is, the following steps are involved in the process of genetic engineering. So we have number 1, insertion of a plasmid into a bacterial host cell. Number 2, use of an enzyme to cut out the desired gene from a chromosome. Number 3, insertion of the desired gene into the bacterial plasmid. Number 4, use of an enzyme to open a bacterial plasmid. What is the correct sequence of these steps? Now, I am going to discuss this in detail because I want you to understand it. Now, this is actually an application question based on genetic engineering, right? Now, when it comes to genetic engineering, this is the modification of the genetic material, DNA of an organism using biotechnology. Now, this usually involves transferring genes from one organism to another so that they can introduce new features or maybe change the ones that are present. Now, a common method involves the use of bacteria and their plasmids. Okay, pause. Why bacteria and what are plasmids? So, bacteria are used a lot in genetic engineering for several reasons. One of the reasons is that they reproduce very quickly through binary fission. So this allows scientists, to easily attain the large-scale production of the proteins that they want. Another reason is that bacteria are unicellular organisms and prokaryotic. So their genetic makeup tends to be simple, so it's very easy to manipulate and change. Now, on to the second question. What are plasmids? So plasmids are small DNA molecules that are present in bacteria. So they are separate from the main bacterial chromosome, as you can see. So what the scientists do is that they take out the plasmids from the bacteria, cut them open using an enzyme, add whatever gene that they want onto it, and then take the plasmid that has now been modified, has been changed, introduce it into a bacterial cell. Either return it into the same cell or into a new cell. Okay, let's start and go in order. First, the gene that the scientists desire, the one that they want, is isolated or removed from the chromosome of a donor. This is done through the use of an enzyme. Now, this aligns with step number 2 in our question. Next, a plasmid is taken from a bacterial cell and cut open using an enzyme. Right? Now, this aligns with step number 4 of our question. Next, the desired gene from the first step, okay, the one we took out before, is then inserted into the plasmid. This aligns with step number 3 of the question. Lastly, the modified plasma is then inserted into a bacterial host cell. This aligns with step number 1. Now, you might be thinking, what are the benefits of such? Okay, why go through all this trouble? I'm going to give you one very important and common way that it has helped us a lot. And by us, I mean humans. And it has to do with production of insulin. Now, insulin is needed for the control of blood sugar levels. But unfortunately, some people either don't produce insulin or don't produce enough of it. And so they need an external support. Now, before genetic engineering, insulin for humans was mostly extracted from the pancreas of slaughtered animals, mainly pigs, yes. And then it was supplied to humans. However, such insulin had a limited supply. I mean, so many people had insulin deficiency and the pigs were not enough to fulfill the demands. Another reason why this was also not well received is because some of the patients had allergic reactions to the insulin from the pig. Okay. So here came the bacteria to save the day. So scientists found a way to isolate the specific human gene that codes for the production of insulin. So essentially, they found the gene in humans that leads to the production of insulin. They took this gene and it was then introduced into the plasmid of a bacteria. Now, this modified plasmid was then introduced into host bacterial cells. So as the bacteria grew and multiplied, the human insulin gene present then started to produce insulin. So after a sufficient amount of insulin has been produced by the bacteria, it is extracted, purified, making it safe for use by humans. It is truly incredible how far science has brought us, isn't it? Here's to scientists, the researchers, and everyone who believes in the power of curiosity and discovery. See you next time.